Hello, everyone, and welcome to the ACS Division of Polymer Chemistry's webinar series. I'm Mike David, and I will be your host for today's broadcast, which will focus on high-impact nanotechnology applications of layer-by-layer assemblies. We are going to hear from two chemists today, Dr. Paula Hammond, who is the David H. Koch 65 Chair Professor of Engineering and Department Head at the Massachusetts Institute of Technology, Department of Chemical Engineering, and the Koch Institute of Integrative Cancer Research. Dr. Jamie Grunlin, the Linda and Ralph Schmidt 68 Professor, Department of Mechanical Engineering at the Texas A&M University, will be joining us as well. Our moderator for today is Dr. Mark Hillmeyer, who is the past chair of the Division of Polymer Chemistry, as well as the McKnight Presidential Endowed Chair at the University of Minnesota. And now, I'm pleased to turn the time over to Mark to get us started.
Thanks very much, Mike. It's again my pleasure to host this now fifth in a series of webinars brought to you by the Division of Polymer Chemistry. As you heard, I'm in the second half of my past chair term, the Division of Polymer Chemistry. And I have to say that I'm excited to host today's presentation and happy to bring these webinar series to a number, I know, of Division of Polymer Chemistry members who are unable to make ACS national meetings, where both Paula and Jamie are routine presenters of their research. So I think it's very nice that they both agreed to be able to present their work on layer-by-layer assemblies in this webinar format. For those of you who are new to the Poly webinar series, welcome. I'll just let you know what's going to happen is we're going to have about 20 or so minute presentations, first from Paula and then from Jamie. And then at the end of Jamie's presentation, we'll be fielding questions. Again, welcome everybody. Thank you to both Jamie and Paula for their willingness to do this. And I'm going to now turn it over to Paula for the first presentation. Paula, please.
Thank you, Mark. I'm really excited to talk to the group today and describe some of the things that we can do with layer-by-layer assembly. I'm going to focus on our work in biomaterials and how layer-by-layer assembly is a very fascinating route for drug delivery. Layer-by-layer itself is a very simple process which involves the alternating adsorption of positively and negatively charged species onto a substrate. So, for example, one might imagine that you can have a charged substrate, something as simple as a glass slide that can be immersed into a dilute aqueous solution. If that slide is negatively charged, then a polycation, or positively charged polymer, can adsorb onto that substrate until ultimately the surface charge is reversed. Upon rinsing, you can remove anything that's not truly adsorbed, and then adsorb yet again another oppositely charged material set, and that will adsorb until surface charge is reversed. And that process is repeated again and again. Now, these layers can be as thin as essentially the molecular cross-section of a polymer chain, just a few angstroms, to as thick as several tens of nanometers, depending on the ionic strength and pH of the solution and the charge state of the system that is being adsorbed. So we have a great deal of room for tuning these materials. The polymers themselves, although we're making these independent layers with each step, are really polyplexes in which there's a great deal of interpenetration from one chain to the next. So you can imagine that a polymer chain that is adsorbed in layer number 4, for example, may actually have chain segments that go down as deep as layer 1 or as high as layer 8, depending on the type of system that you incorporate. And although you can use alternating charge to build these films, you can also use other kinds of secondary interactions, such as hydrogen bonds, and introduce hydrogen bond donors and acceptors as well in these kinds of layer-by-layer assemblies, along with a number of other interactions. Now, the reason that we're interested in this process for biomaterials is that it's a water-based process, and we can introduce a broad range of different biologic drugs, including proteins and nucleic acids. Now, you can imagine a more traditional drug delivery process. You might use something like a polyester. And the contrast here is that many of our interesting degradable polyesters are only soluble in organic solvents. And those solvents can actually degrade or deactivate a number of proteins. And if we have to add heat to that to get a good solution, then you can also destroy your active component. On top of that, it turns out that the rules of polymer science, of course, are that you can only put a small amount of this biomacromolecule, for example, in the polymer before you get microphase separation. And once you get that phase separation, you're going to have a more bolus release. Finally, if you introduce multiple components into the film, you end up with all of them coming out in the same fashion. One of the nice things about layer by layer assembly is that if we have a nice set of, let's say, degradable polymers that can be incorporated into the film based on charge, for example, these poly-beta amino esters, which are shown in the upper right-hand corner, which have ester groups that are degradable, but also these nice charged groups, you can tune the degradability of these polymers and then build up films by alternating them with the active agent. And in our case, those active agents may be proteins, nucleic acids, or even small molecule drugs. So you can imagine that you can incorporate different drugs with different degradable polymers, and they will each have their own unique rate of release from the film. On top of that, we get beyond that limitation of drug loading. Rather than having just a 1 or 2 weight % loading before phase separation, here the thermodynamic rules are essentially charge compensation. So we can get loadings that are as high as 30, 40, or 50%, depending on the architecture of the film and what we're absorbing. So we can get very compact and dense amounts of material into very thin films. Finally, if we're able to introduce very nice diffusion barriers between the layers, then we can get not only unique release profiles, but phased release in which the drugs come out at different times. And these can be very advantageous if we want to have a more complex release profile in which we release, for example, something like an antibiotic followed by a growth factor. We've been doing a lot of work, in fact, looking at these systems for bone regeneration. And an example of this is the use of BMP2, which is a growth factor that stimulates the stem cells in our body to generate osteoblasts. This is actually a factor that can be very potent. Now, one of the things that we have right now out in industry is a sponge, a collagen sponge that releases large amounts of BMP2. But unfortunately, because there's not a real control of that release, we end up getting a bolus release of BMP2 in the body. What this means is that for something like a regenerative approach for spinal cord injury, for example, a large amount is released over a very short period of time. A little bit remains behind at the base of the spine and does its work regenerating bone, but a large amount of the growth factor is distributed through the bloodstream and can actually activate nascent tumor cells or grow bone in places where it doesn't belong. And for this reason, the current product, it has a black box warning from the FDA. However, BMP2, although it's a potent molecule, is a very productive one that when used in the right amount can essentially be life-changing in terms of its ability to regenerate bone. We're also interested in other kinds of growth factors, ones that can help build the vascular system, like these two here, vascular endothelial growth factor or platelet-derived growth factor, both of which help support the development of the vascular system. When we generate these growth factors into layers, we treat these proteins as positively charged species, essentially as polycations, because their isoelectric points are 8.5. So at pH 7, they can be treated as weakly positively charged materials that can be incorporated into a film that has a tetralayer composition. And in that film, we have essentially a polycation that degrades, like the ones I showed you before, poly-beta amino esters, a polyanion, which can be polyacrylic acid, or polysaccharides or glycosylaminoglycans that are negatively charged. And we have our protein sandwiched between that polyanion. We can actually use this to address a number of different issues, such as large bone defects that are so large that they cannot heal. For example, in this work, which was done by my former graduate student, Nisarg Shah, he actually looked at the coating of membranes, which allowed us to essentially cover these large cranial defects and regenerate bone. This is in place of the traditional approach, which is to harvest bone from another part of the patient's body, such as the hip, grind it up, and then try to regenerate bone from the cells and material generated from that graft, and use something like an artificial implant to help support that. And of course, there are a number of problems with both the sacrificial bone and the introduction of a foreign material into the skull. In our case, we've been using a degradable polylactic glycolic acid membrane, which is generated using a very standard phase inversion approach. It's porous and creates a surface on which we can then build our layer by layer film. The membrane then acts as a physical and mechanical support that degrades, and our layer by layer thin films coat that support, and without changing its mechanical properties, can release potent amounts of growth factor. So here is an SEM image showing the porous scaffold in close-up and our layer by layer film on top of it. And what you can see is on the right, the release profile that we got when we essentially introduced BMP2 layers of this layer by layer film, and then introduced these platelet-derived growth factor, or PDGF, layer by layer films on top. So BMP2, as you recall, is the bone-generating growth factor. The top is the PDGF, which is going to help build the vascular system. And when we actually look at the results, we use a rat cranial defect model. So in this case, we generate essentially these holes in the cranium of rats at a size that is so large that it cannot heal. So this first column shows the untreated animal, and you can see that at no point do we get true bone generated in that defect. In micro CT, we can see the generation of bone in a membrane that is coated with a layer by layer film that only contains the BMP2 at 0.2 micrograms. And we see that we're getting regeneration by the 4-week point. If we increase the dose to 2 micrograms, we also see regeneration. These are actually considerably low doses. The typical dose that is used in the collagen sponge product I described is on the range of 3 or 4 milligrams as opposed to micrograms. Now, we also see that if we introduce BMP2 at 0.2 micrograms and the platelet-derived growth factor, 0.2 micrograms, that combination film gives us a somewhat faster healing, and that we can see here. But it's only when we look at the actual histology of the bone that we see the impact of introducing the dual growth factor system in which we have staged release. So here we have a membrane that was placed across the cavity, and all of these are histological slices in which we can see the bone margins on either side, a critical defect that did not heal in the untreated case. With just the membrane, we get a collagenous membrane that is generated across it, but no true bone. We do get very nice solid bone generated at 0.2 micrograms, and at 2 micrograms, we see even more deposition of bone. The problem here, though, is that we see a lot of defects in the bone, lots of gaps, but also roughness along the edges. It turns out that the mechanical properties of the bone with just BMP2 is actually weaker than that at the margins, both with respect to impact strength and with respect to modulus or stiffness. However, when we introduce PDGF, which supports vascularization, and if you remember from that release curve, PDGF came out over the first 7 days, and BMP2 was released much more slowly over a period of 30 days. In this case, we see that we get a highly vascularized bone, and that the bone that is generated looks very much like the bone at the margins. On top of that, we see that the mechanical properties match the native bone, high impact strength and modulus. And we think what happens here is that we are supporting first the generation of blood vessels across this material matrix, and second, the recruitment of these bone progenitor cells, which then begin to deposit mineral around the vascular system. The fact that we have blood vessels not only provides a more oxygenated and healthy bone, but also promotes the progression of osteoclasts, which are the cells that remodel tissue, to arrive and remodel the bone so that we get this very nice, highly oriented bone in this case. So I have a little question for the audience, and I'm going to turn things over to Mike.
All right, our first question for all of you is, which of the following cannot be incorporated into a layer-by-layer film? Is it the antibodies, the microRNA, the collagen, the disilophenolic, or none of the above? And thank you for putting in your responses. Seems to be pretty one-sided on this, so I'm going to be closing this poll in 5, 4, 3, 2, 1. Well, 56% of the audience said it was none of the above, which was the majority by far and away. Second highest was collagen with 19%. And with that, I'll turn it back over to you.
Excellent. Thank you, Mike. And it turns out that the audience was correct. None of the above. All of those species that were listed can be incorporated. And layer by layer, we can incorporate large macromolecular charged species. We can incorporate small molecule charged species and natural biomolecules like collagen. But we can also incorporate drugs like diclofenac and other hydrophobic drugs by using a carrier. We can get help from something like a polycyclodextrin, such as the one that's shown here. You can see that these cyclodextrins, which have hydrophobic pockets, can sequester hydrophobic drugs. And there are a number of sulfated polycyclodextrins that are water soluble and can be layered into these films to allow incorporation of even these hydrophobic species. All right, I'm going to give a very brief second example of layer by layer from a large surface. We can also release siRNA directly to wounds using the same kind of approach. And the reason that we might want to do this is to correct the overexpression of specific genes that might occur in wounds because of some sort of change in the metabolic function. For example, for diabetics, chronic wounds are a common problem. And in these hyperglycemic patients, there is often overexpression of the collagenases that break down tissue at early stages. One of these collagenases is MMP9. It actually breaks down collagen, and at early time frames when we really need to be laying that collagen down to generate a matrix for wound healing. So we actually incorporated siRNA to silence this MMP9 so that we could improve the wound closure process in diabetic patients. This is the work of my former student, Stephen Castleberry, and in this case, he coated layer by layer films that contain siRNA in alternation with a very simple and fairly common polysaccharide, chitosan, which is shown here on the left, has a positive charge to this primary amine and is derived from crab shells. Chitosan complexes siRNA very effectively, but it is not rapidly degradable. So he actually created an underlayer of a layer by layer film that contains the degradable polybeta amino ester, poly2, and dextran sulfate, which is really a benign negatively charged polymer. This underlayer is meant to break apart and allow the release of the siRNA containing chitosan layer. The chitosan protects the siRNA for long periods of time in the wound cavity. We're able to directly coat nylon dressings using this layer by layer approach. Here you can see different loadings. We can see the linear growth of these films, and we can actually tune how much we deliver using this approach. And using a simple diabetic wound model, this is a diabetic mouse model of a wound in which we are looking at both a treated and an untreated control. We're also looking at a layer by layer film that contains a scrambled siRNA. And what we can see is that the amount of expression of this gene, MMP9, is decreased in 1 week by about 60%, and in 2 weeks by as much as 85%, indicating that over extended periods of time, this siRNA, which normally is rapidly degraded in the bloodstream, and certainly rapidly degraded in the rather low pH and high enzyme environment of the wound, is in fact sustained for extended periods of time. And what we see in terms of wound healing is that after 7 days between the untreated and the treated, which is on the top, we see that the treated sample has a narrow bridge of collagen, which is stained in blue here. After 14 days, we see that that narrow bridge has become a large sort of chunk of tissue that has been deposited in comparison to the untreated wound. So we can see also effective increases in the rates of wound closure in these animals, both with the PC muscle and the epithelial closure. Okay, a quick question from Mike again.
All right, for our second question is, what is the smallest system coated with LBL? Is it a mammalian cell, a single atom quantum dot, a virus particle, or a gold nanoparticle? And with this, I'll be closing the poll in 5, 4, 3, 2, 1. Well, Paul, this was a little bit more split. Gold nanoparticle slightly won with 38%, and atom quantum dot was coming in second with 33%.
Excellent. It turns out that the smallest system that's been coated so far that's been reported is a gold nanoparticle. And some of the smallest have been between 5 and 10 nanometers. This is actually rather fascinating because these polyelectrolytes can wrap around extremely small nanoscale material systems. And I'm taking this image here just to give you an idea of this resolution from one of the papers by Schneider and Decker from 2008, in which they actually talk about the conditions which allow these polyelectrolytes to wrap around nanoparticles based on the hydrodynamic size and the degree of extension of these chains. Clearly, it's going to be sensitive to things like pH and ionic strength. The fact that we can coat nanoparticles is actually key to the third example that I have today, which is the coating of nanoparticles for targeted drug delivery against tumors. In this case, we're actually trying to address very aggressive tumor types in which we have a gene or genetic modification in these tumors, which allows the tumors to survive even in the presence of chemotherapy drug. So in this case, we incorporate our chemotherapy drug in a nanoparticle that is familiar to us. For example, we can use a liposome, we can use a PLGA nanoparticle. As long as that core particle has charge, negative charge, which is what we use most frequently, then we can layer around it. So we wrap up our drug in the core, and then we absorb around the core layer by layer film. And in our case, we typically look at 2 to 4 layers. We can absorb a positively charged layer and then introduce siRNA, which is going to essentially silence the gene that allows these tumor cells to survive. So the idea is to lower the tumor defense mechanisms with this silencing of the gene. Then we're going to use another positively charged layer, and that's going to completely encapsulate our negatively charged siRNA molecule. Finally, we need to get this nanoparticle to distribute through the bloodstream. It's a systemic exposure that requires that the nanoparticle not carry positive charge, because positive charge will engage with the cell membranes of many of the cells in the bloodstream. So we're going to wrap it with a negatively charged nanoparticle, and we'll choose one that has a great relationship with water, because we want this to be a stealth layer that carries a large number of bound water molecules to it so that it does not tend to essentially attract the attention of the monocytes, the immune cells that travel in the bloodstream. So there's a lot of engineering design that goes into this approach, and we also rely on several mechanisms to get our nanoparticle through the bloodstream and into the tumor. And one of them is simply the use of size. It turns out that for a number of tumors, especially at the early and moderate stages of tumor growth, a lot of vasculature is created rapidly, and that vasculature will tend to have defects in it. There's a general belief that this leakiness of the vasculature will allow nanoparticles that are going through the bloodstream to leak into the tumor when they reach the tumor blood vessels. And that allows these nanoparticles to essentially get into the tumor tissue and reside in that tumor tissue for some time before they find another leak to get out again. So I consider this kind of a pinball machine effect. And the idea here is that it gives us an accumulation of the nanoparticle for extended times. And here you can see these mice with tumors on their backs. And after a period of about 24 hours, we can image the quantum dot that was coded in this case and see that these nanoparticles have accumulated in these mouse tumors. Now, that's just one way to get into the tumor. And of course, if we do get in, we have to worry about whether or not tumor cells will actually take up the nanoparticle. And because we've been very careful about disguising these nanoparticles as essentially water-shrouded objects, we need a reason for these nanoparticles to be taken up by the tumor cells. It turns out that one of the mechanisms by which we avoid these interactions with healthy cells is through strong negative charge, and the other is through the shroud of water that we have. At pH 7.4, our nanoparticles have a strong negative charge of about minus 30 millivolts. And we have these fairly well arranged nano layers, as you can see on the image on the far right. What we have found is that when we use layer by layer films, we can use the unique capability of combining acidic and basic groups to generate nanoparticles that have an isoelectric point, that these nanoparticles can actually shift in charge as we begin to protonate the carboxylic acid groups that may be in the outer layer. And that allows us to essentially lose some of that negative charge. It also erases some of the ionic crosslinks and the film swells. So at a pH of 6, for example, we have a relatively neutral nanoparticle that is soft and squishy because of the swelling of the layers. And it turns out that cells will much more likely take up these neutral nanoparticles than the strongly negatively charged water-shrouded ones. So we can take advantage of this because tumors tend to be hypoxic. They're not great at getting oxygen to their core, and so their pH is somewhat lower. And in those hypoxic conditions, the nanoparticle transforms into a sticky nanoparticle, and we therefore can get enhanced uptake. And we can see this when we, for example, take a section of a tumor and we stain it for hypoxia, which is shown here as the green stain. And we stain for our nanoparticles. Our nanoparticles here have a fluorescent tag, and we can see the overlap in yellow of our nanoparticles in hypoxic regions. Now, this is in the case of tumor cells in which we have not targeted a specific marker for the tumor itself. But it turns out that many tumor cells overexpress a given protein on their surface. And we can use that as a third way to target our nanoparticle to the tumor cell. In our case, it turns out that we have been using hyaluronic acid, a nice negatively charged polysaccharide that is present in our body as part of the matrix of our tissues, as the outer layer. Hyaluronic acid, or HA as we call it, is very highly hydrated and strongly negatively charged, and it has a large number of carboxylic acids on its surface. So we can essentially take advantage of the effect I described earlier of hypoxia to make these actively taken up by tumor cells. But it also turns out that hyaluronic acid binds a receptor called CD44, and CD44 is highly overexpressed on a number of the solid cancer tumor cells that we are targeting, including those in triple-negative breast cancer, non-small cell lung cancer, and ovarian cancer. So here we can see some cells which have taken up our nanoparticle, which is shown in red fluorescence, and the case where we essentially titrate away those interactions with excess hyaluronic acid and eliminate that uptake. We can also see a tumor slice in which we stain for CD44, and we can see the nanoparticles and the overlap here in the tumor section in yellow. We get a very high accumulation here. This gives us three ways of targeting the tumor: by size, by tumor hypoxia, thanks to these layer by layers and their tendency to be responsive to pH, and by cell receptor through the choice of that outer layer. Finally, of course, we have to incorporate our cargo, and our cargo is siRNA, which is going to silence one of the genes that allows these tumor cells to avoid death. And to do this, we had to optimize against a range of different polycations. And this is a short list of some of the polycations that we used with our siRNA. And we looked at loading, but we also looked at whether or not we were introducing toxicity to the nanoparticle. And it turned out that poly-L-arginine, a simple homopolypeptide, is a great system for not only incorporating the siRNA in sufficient amounts, but allowing us to get the nanoparticle both in the cell and out again from the endosome into the cell cytoplasm, where siRNA can be translated. This is also the direct result of buffering effects in our layer-by-layer film, in this case induced by the presence of poly-L-arginine. And the way that it works is that the nanoparticle, when it's first taken up in the cell, is taken up in an endosome, a vesicle which has a pH which is getting lowered from 7 to 6.5 to 5.5. And as the pH is being lowered in this endosomal compartment, essentially we're seeing the opportunity for these nanoparticles to also undergo buffering. And in that buffering process, they actually create some small leaks in the endosomal compartments, and we get endosomal escape. And you can see this at later times, that the nanoparticle is dispersed throughout the entire compartment of the cell, except the nucleus. Once we get out in the cytosol, the siRNA can be freed, and essentially it gets to load onto the RISC complex and silence. Finally, a simple demonstration of this was to target a known target. This one is called MRP1, and it's a pump. It essentially sits at the cell membrane, and when a toxic drug, like a DNA damaging agent, enters the cell, it gets pumped out right away. I think it's a kind of a crazy mechanism that tumor cells have for avoiding cell death. What we find is that we can silence it. So we introduce siRNA against that MRP1 pump. We incorporate a known DNA damaging drug in the core of our nanoparticle. And then we look at that nanoparticle, which now has the chemo drug and siRNA, and we introduce injections over time in mice with established xenografts. If we just look at the silencing of the MRP1 pump in these tumors, we can see that we're getting about 85% silencing determined by qPCR and by staining. If we look then at the efficacy of therapy, I'll just focus on the right-hand side here. You can see that we have our saline control, in which we have rapidly growing tumors. However, and we can see in the middle that if we just incorporate doxorubicin with a scrambled siRNA that doesn't encode for anything, that we can essentially get some efficacy, but the tumors are still growing at a relatively rapid pace. And we can see this in the data here quantitatively with the controls in green and black and the doxorubicin in blue. Finally, if we look at the combination system, we actually see that we've not only stopped the growth of the tumors over time, but we're seeing a decrease in the size of the tumors when we're able to silence that MRP1 pump and then administer that doxorubicin drug from the same nanoparticle. So we've moved on to more difficult targets, and I won't have as much time to talk about this, but we can combine targets. For example, in non-small cell lung cancer, we can combine siRNA against a known oncogene called KRAS, and we can address the lack of P53 through the introduction of a microRNA that replaces the function of this lost gene. This lost gene is one that actually protects against cancer. So we can combine multiple nucleic acids into a single nanoparticle, as well as a chemotherapy drug. In this case, we can look at the siRNA and microRNA coming out rapidly and the cisplatin coming out more slowly. And the net result in a realistic model in which we generate lung tumors in what's called a syngeneic model, we can actually see accumulation of our nanoparticle in the lungs of tumored mice compared to healthy mice in very high concentrations due to the presence of CD44. And ultimately, we can see that we can extend the lifetime of those mice significantly with our combination nanoparticle compared to a nanoparticle that just contains, for example, cisplatin alone. So I talked about this layer-by-layer approach, both on a macroscopic layer level and on a microscopic level, and finally on this nanoscopic level with targeted nanoparticles. I think it's a very modular tool that can be used to address a very broad range of delivery questions when we think about delivery from a surface. And we can use the modularity of the approach for combinations and sequential release. And finally, I'd like to thank the students who did the work and funding and the research lab in general. I'll turn things over now to Mark. Thank you very much.
Paula, thank you very much. We're going to take this moment to switch over to Jamie's presentation. And while we're doing that, what I'll just remind you is actually been getting many good questions online for Paula. I've been curating those and getting them all ready for the Q&A session after Jamie's talk. So please do continue to use that online portal for questions. Now I'm going to turn it over to Jamie Grunland at Texas A&M University. Jamie?
Thanks for having me. It's a pleasure to be here, and it's an honor to follow Paula, who's a superstar in this layer-by-layer field, and that was very interesting, and it takes some of the burden off of me in this presentation. I'll talk about layer-by-layer, but going maybe in some other non-bio directions, I suppose we could say. And so in my group, we do a number of things that I would call them all coating related, and three of the most prominent properties that we focus on are flame retardant treatments and gas barrier and thermoelectric nanocomposites, but I'm going to focus on these first two briefly in this seminar. So you can see my outline here, and I've underlined the key points, so I'm not going to read those to you. Paula showed a schematic of how the layer by layer works. I often show this little cartoon. It's actually as simple to build these films as it sounds. If you have a substrate, in this case I'm showing it negatively charged, but it might be uncharged or it might be cationic somehow by some functionalization, but regardless, that substrate could be a fiber, piece of plastic, film, metal, glass, doesn't matter, silicon wafer. In this case, you're looking at a cationic polymer being layered with a anionic plate of some type. In my mind, that's a clay platelet, which is the basis for a lot of the things I'm going to show you today. But anyway, it's a very convenient coating tool. You can process under ambient conditions, but get properties that are like something that's been chemical vapor deposited. Here you're looking at a cross-sectional TEM image that looks like maybe something you found in your shower drain, but it's actually 40 layers of clay with polymer in between. So we would call that a 40 bilayer polyelectrolyte film. And so in my group, we first started working on this when I first became a professor on gas barrier layers. And you can see a snapshot of some of the publications we have in that topic. And we're interested in many of the things that probably all of you are interested in, and sometimes maybe you didn't even think about the barrier layers that are involved in some of these different applications, like the flexible displays, which you need barriers to protect the electronic components in those displays. So barrier is very important across all these applications. The established, especially when you're talking about transparent gas barriers, you're talking usually about metallized film or something like a silicon oxide that I'm showing here, or a bulk composite that somebody put clay or some other nanoparticle in to stop gas. One of the problems is you can get cracking in the silicon oxide. You can also get a lot of opacity because of aggregation in the bulk composite with that clay, as I'm showing here as well. So with layer by layer assembly, I always tell people I kind of get all the benefits of these competitive technologies without their drawbacks. I don't suffer from these same issues that they have. And so a perfect example is polyethylene imine, which I'm showing as PEI here. pH is a subscript in green there, or in some kind of a green. And then MMT is montmorillonite clay platelets that are something like a 200 nanometer diameter, 1 nanometer thick. But when I layer that cationic polyethylene imine with montmorillonite clay, here it's all 40 bilayer films. That means 40 clay and polymer layers. What happens is my oxygen transmission rate is decreasing as my clay spacing is increasing, and I'm actually influencing the clay spacing with the pH of the PEI that's being used. If I increase the pH of PEI, I'm decreasing its charge density. That means it's a weak polyelectrolyte, and it deposits more thickly because it's not repelling itself during the layer by layer assembly. And so we can really dial in a gas barrier, in this case an oxygen barrier, and it's because we're building this very intricate nano brick wall structure that we call it, and you have a very tortuous path for any gas molecule that's trying to get through there. Even hydrogen barrier can be created in this fashion. And so when we knew that more polymer between the clay in some cases was helpful up to some point, we started to put more layers. And in this case, you're looking at a polyethyleneamine, polyacrylic acid, polyethyleneamine, montmorillonite clay quad layer. Paula referred to it as a tetra layer. They're both cute names, I think, for this structure. But anyway, you can see in my cartoon, I'm actually showing that it's getting a little thicker with each quad layer deposited. And I guess first I'll show you how we make this in the lab. Commercially, you could make this using some kind of a roll-to-roll spray coating or dip coating or blade coating type process. But in the lab, we just use a dipping robot that will go down, and in this case, the cationic polymers are on the left, and the clay and the polyacrylic acid, the anionic materials, are on the right. And it's just taking this, in this case, piece of PET or polyester film through a 4 dipping sequence, and then it's rinsing and blow drying in between. You don't necessarily need all the steps I'm showing you here. This is kind of the most academic way to make a film. And so anyway, when we look at the growth, which I can use ellipsometry to measure the film thickness because these are transparent films, if I deposit it on a silicon wafer, you can see that the growth is very exponential for this system. If I only looked at polyethyleneamine and polyacrylic acid, it's actually even more exponential because those are 2 weak polyelectrolytes that are highly interdiffusing with each subsequent layer deposition. The clay slows that process down to some extent, but you're still getting some interdiffusion and therefore what sometimes is known as exponential growth. So you can see how film thickness is increasing and clay concentration then is decreasing because I'm getting more polymer between each of those clay layers. So here's a 5 quad layer film. This is less than 100 nanometers thick, and you can see it's completely transparent. That's on top of that PET film there. And when we look at oxygen transmission rate now, just 4 quad layers gets me what we would call an undetectable oxygen barrier. This is measured by a tool called a MOCON permeation instrument, and it's well known in the food packaging industry. You can also see in this TEM cross section how much more spaced out the clay is relative to that first image I showed you that was just a bilayer with a single polymer layer between each clay layer. We got a question now, so I'll turn it over to Mike to hand it over to us.
What is this nano brick wall made of? Is it very small bricks and mortar, exfoliated clay nanoplatelets and polyelectrides, calcium carbonate and protein, or polystyrene and polyethylene? And I don't think anyone is really doubting this too much. It's very much a runaway, so I'm going to close this poll in 5, 4, 3, 2, 1. Well, 88% of the audience said it was exfoliated clay nanoplatelets and polyelectrolytes, and the next highest was 9% with polystyrene and polyethylene. So with that, I'll turn it back over to you, Jamie.
Okay, I'm assuming that was the polymer lovers that put polystyrene and polyethylene. Yeah, it is in fact exfoliated clay nanoplatelets and polyelectrolytes. The calcium carbonate and protein is kind of the abalone shell structure, which is a brick wall type structure, but it's more of I'd call it a micro brick wall as opposed to a nano brick wall. So what we're seeing here now, I had mentioned putting these layers between the clay, and some people over the years had asked me if I only took a look at that polymer system, the polyethyleneamine with the polyacrylic acid, what would happen in this case if I tested its gas barrier? And I assumed it would be very bad because it's just a completely polymer thin film. What we did see, though, is when you combine polyethyleneamine and polyacrylic acid, you've got a polyacid and a polybase, and they really love each other more than they love themselves. So you actually get a higher TG than even what an upper bound, a wood type of combination of those two TGs would be. And you also get a pretty high density film, much higher than a typical polymer. So it could be something like 1.3 or 1.4 gram per centimeter cubed. So it's very unusual interaction, but high, strong interaction between these two polymers. Sometimes it's referred to as a scrambled salt structure, which you could think of as interpenetrating network on steroids or something like that. And it turned out that an 8 bilayer film, I can tell you that's about 600 nanometers thick, had an undetectable oxygen transmission rate. So now here is without any clay. Now this is 600 nanometers, and I needed only 50 nanometers with a clay film to get that same high barrier, but it's just demonstrating once again the power of these ionic interactions. And there's also hydrogen bonding in this case as well. But anyway, there's such strong interaction between these polymers. And then once you cross-link it, we can maintain this very high barrier even at 100% relative humidity. In fact, that 10 bilayer film is not even cross-linked, and it's about 1 micron thick, and it's undetectable gas barrier even at 100% relative humidity, which could be great for something like food packaging. Many people, though, say, well, 10 bilayers, that's 20 layers, that's a lot. And so there's a number of things we've done to reduce the number of layers we need for high barrier. We found from another group that they had added this tris buffer. And when they added that with the polymer, the cationic polymer, they got a thicker growth with clay. And it was because there's some kind of complexation between the tris buffer and the clay in solution. And so you can see in our case, we took chitosan, and Paula had showed the structure of that chitosan earlier. Chitosan plus the tris buffer in one solution. You use a tris rinse, meaning tris in water, and then vermiculite clay in this case. That's a very high aspect ratio clay platelet, something like 2 microns in diameter. But you can see how the thickness we get. 6 bilayers is almost 2 microns thick. 8 bilayers is about 4 microns thick. And you can actually use an optical microscope to see this layer by layer assembly, which this is a super, super thick layer by layer film. And so if I look at oxygen transmission rate, just a 4 bilayer film had this 0.122 in these units, cubic centimeter per meter squared per day. The 5 mil PET substrate that we were using, that's almost 100 times improvement over the 5 mil PET, which is 125 microns thick. So you add 1 or 2 microns and you get 100 times improvement over 125 micron film. So 4 bilayers is 8 total layers. And we have a number of other tricks we can do too. So this is just highlighting one example. It turns out that as a segue to the other little part of my much shorter part of my talk, in this case, we took a 3 millimeter thick polystyrene plate, and on one of the plates, we actually put an 8 bilayer thick layer by layer assembly, and you already saw the video there. The polystyrene with no coating on it, of course, is highly flammable, and it'll completely burn up. That 8 bilayer coating was 4 microns. It's a negligible thickness. You couldn't even measure the weight gain on that plate. And yet I can hold a torch there for 15 minutes and not burn through that very flammable polystyrene sheet. So that highlighting kind of some of the fire resistance things we can do as well. And so Chemical and Engineering News 5 or 6 years ago, I think it was 6 years ago, had an issue where the whole cover issue, most of the issue was focused on flame retardants, and it talked about toxicity of flame retardants and what should we be doing about the future of flame retardants in general. And they kind of ended by talking about some new opportunities for quote unquote safe flame retardant. And they highlighted this layer by layer flame retardant technology that we've been working on. And so that was good exposure early on in our efforts. And we very recently, last year, published a review. So the last decade, it's only been about 10 years of research on this topic. It's a subfield of flame retardant chemistry. So if you're interested, you can check that out later. And of course, we're well published, and we've gotten, somehow I've gotten a lot more interest in this topic, I guess, because it's much more safety focused. But anyway, it's been a fun topic to work on, and all the kids in my lab want to work on fire retardancy because they like to light things on fire. But in relation to fire, here's a question that Mike's going to give to us.
Just testing some real world knowledge. In the event of a fire in your home, you should immediately run to a pre-planned meeting spot away from the home, Search for others around the house to ensure their safety before leaving. Call the fire department or wait a few minutes to see if it self-extinguishes. And again, everyone's voting pretty quick, so I'm going to close this in 5, 4, 3, 2, 1. And Jamie, we had 55% of the audience say go to a prearranged spot, and 31% say call the fire department. And those were the highest too. So with that, I'll turn it back over to you.
Okay, yep. So the first thing you should do is the winner immediately run to a pre-planned family meeting spot. I don't have time in this presentation to show the video I have, but once your home catches fire, if a couch starts fire for some reason because of a candle or a cigarette or something like that, you literally have 2.5 minutes to get out of your house. And probably by the time you even realize there's a fire, you have about 30 seconds or something like that. So you don't have time to look for anything, grab anything. If you want to live, you will leave the house immediately. Then you can call the fire department or do anything else you need to do. So, of course, we would like fires to be stopped. And so there's this intumescent concept, and a lot of skyscrapers, the steel beams in a skyscraper have this intumescent kind of white, thick coating on the steel beams. And when there's a fire, it puffs out, and you can see a thickness of 10 centimeters down in this corner picture. That's on a piece of steel. We wanted to do the nano/micro version of this using layer by layer assembly. And so what we did is we layered this nitrogen-rich polyallylamine with this phosphate-rich polysodium phosphate. And then we put this on cotton fabric, and the cotton itself was a carbon donor, the phosphate's an acid donor, and the blowing agent, quote unquote, that bubbles things up is the nitrogen-rich polyallylamine. And in the lab, this kind of technology is actually maybe even more simple than some of the other things I've been showing, because the textile world uses pad dry processing, which just runs fabric in and out of liquid baths anyway. Here, so basically my student here is effectively doing pad dry processing by hand to build up layer by layer the coating. And so what we can see here is uncoated control film and a 20 bilayer coated polysodium phosphate polyallylamine. It's a lot of layers, but anyway, what I want you to notice here is, I hope you can see a difference between these two fabrics. That uncoated cotton, of course, is extremely flammable, completely combusts 100%. And then this coated fabric passes the standard fire test, which is this ASTM 6413. It's a vertical burn test. And you can even, with your naked eye, see kind of this micro intumescent effect. So we're getting what you get on the steel girder in a skyscraper, just on a very micro scale. So we've of course patented this technology, as you might imagine, and we work with various partners to use this on textiles, but of course on other substrates as well. Turns out cotton's easy to flame retard. Most of you are probably wearing some blend of cotton with nylon or polyester or something like that. So that doesn't flame retard as easily. That's more complicated. And so what we did was we put chitosan and melamine together in one solution and polysodium phosphate in the other solution. And we did layer by layer with that. And when you put that onto polyester cotton fabric, it doesn't pass the vertical flame test if it's just the chitosan with the PSP. So that would be something that would flame retard cotton, but it will not flame retard polyester cotton. Then here's 12.5 weight% of this chitosan melamine and polysodium phosphate. And that does pass the vertical flame test because of that addition of melamine, which has some kind of a cooling mechanism, and it interacts much better with the polyester. It's true also of the nylon. A recipe very similar to this can flame retard nylon. And I should mention, most of the recipes you're looking at have been further improved from what I'm showing here. So there's fewer layers to get the same result that you're looking at here. So I'll just finish up by showing, even in my own lab, we built a little pilot coater. This is basically what you would call a pad dry processing equipment. Film goes in and out of liquid deposition baths. I know that there are companies, and even Paula Hammond's own research group created a technology of spray coating layer by layer assembly. So there's 5 or 6 different water coating methods that you can use, some very traditional ones, to deposit roll to roll layer by layer assembly. This is cotton fabric on a on my little pilot coater, and then we built a bigger pilot coater that's a 50-foot, 12-inch wide coater, and we coated thick rubber, thin PET film, fabric. We've done a number of things, and it works very smoothly. So I'll acknowledge my research group, and I appreciate everybody listening. Thank you.
Jamie, excellent. Thank you very much. Thanks to both of you very much. I love being not only the moderator of this session, but also learning from the presenters, and I learned a bunch in this case as well. We have been getting a number of questions coming through on both presentations. I'll start with Paula. Specific question on siRNA aspect of your talk, and Sunil says, how would you be able to control the loading capacity and quantify the loading capacity of a macromolecule like siRNA in the work you talked about?
That's a great question. We actually use several methods for determining the siRNA loading, but they're actually stains and assays that you can do for siRNA. We will use a colloidal suspension in which we know the exact amount of material that we have and determine from that the siRNA. So we typically measure it in something along the lines of micrograms or nanograms per milligram of our formulation. We've also done the back calculations based on the size of the nanoparticles that we use, which are around 100 to 120 nanometers in the nanoparticle systems, to determine that there are about 2,000 to 3,000 copies of siRNA per particle. In the case of the nylon mesh, it's even simpler because you have essentially the ability to coat a planar surface with a defined surface area and a defined number of layers. And we change the loading in all of these cases by increasing the number of adsorption steps that we introduce of siRNA, and it increases for us pretty much linearly with the number of layers because siRNA is so strongly charged that it's in this sort of linear growth regime.
I see. Well, thank you. Again, a specific question for you, Paula. This is from Justin. Are there specific advantages of poly-L-arginine over poly-L-lysine as a cationic layer in the LBL particles?
Yes, actually, it turns out that poly-L-lysine is not as effective at allowing our siRNA nanoparticle system to get out of the endosome. We think that when we deliver these nanoparticles, when they get incorporated into the endosome, that they undergo some buffering activity. We also know that they undergo the layer by layer film undergoes some swelling. When we have poly-L-arginine, we believe that we end up shifting to actually a positively charged polymer, and that positive charge on the surface helps to compromise the membrane integrity of the endosome, causes leaks, and allows the compartment to enable siRNA to leak out. Whereas poly-L-lysine, which has a higher pKa, we don't see quite the same effect.
I see. Larry asked, how is the adhesion between LBL layers and the PLGA membrane? Do you need to do any surface treatment on the membrane in order for that first layer, I guess, to have good adhesion?
Yes, in fact, for all of our work in which we coat something plastic, we have typically plasma treated the plastic very briefly, sometimes for as short as 10 seconds or 20 seconds, with an oxygen or air plasma to generate negatively charged groups on the surface. Both for the nylon mesh and for the PLGA membrane, we've done that.
I see. Okay, thank you. Jamie, I'm going to switch over to a few questions for you. They're still actually coming in as we speak, and there was a thank you for showing the video. So I'll ask a question about the video that you showed. I guess those videos were in real time. The first one that you showed with the robot.
All of the videos that I showed were real time. Nothing. There was a hand deposition on the fabric that was sped up, but all the others were real time videos. Yes.
And given that, and we saw that in the very last few slides about your roll-to-roll processes, we have a question from, and I'm going to spell this name because I have no chance of pronouncing it, OLUWATIMILEHIN. What kind of rates can be achieved with the LBL roll-to-roll processes do you expect?
Yeah, so if I'm thinking about practical application of LBL and not an academic exercise, we know that even 1 second exposure time, especially in the case of polymers, particles, maybe it depends, but 1 second exposure times, I've made gas barrier films, for example, very successfully with 1 second exposure to each of those deposition solutions, which means you could run technically at 100 feet a minute, 200 feet a minute.
I see.
Roll to roll.
So this is related to a question by Sujay, which is that this seems like that's fast enough to be practical in some applications.
Absolutely. There's a lot of interest, and of course, I'm sure Paula and I would both say we're open to collaborations with companies on our various technologies, because this is not a crazy esoteric technology. It may have seemed like it once upon a time, but you can make films very quickly, very reproducibly, and very easily with this technology.
Absolutely. I would agree with, on top of Jamie's comment, that this is something that has been proven to be able to take on a commercial market. So I think we're both excited because of the translational capability.
Great. For those investors on the phone, please call them immediately. I have a question about the cotton blends. You made a passing comment, Jamie, and this is also related to Rihanna's question and a question from Paul. Why are the cotton blends more difficult to coat? You made a passing comment about that.
So the cotton and nylon and polyester all burn differently. They're different chemistry. They're all organic. Of course, cotton is cellulosic, so it combusts. It doesn't melt and drip. Both nylon and polyester burn hotter than cotton, and they melt and drip. And so just the chemistry, the way the chemistry of the coating interacts with those different fiber chemistries makes it much more challenging, because I'm trying to fight off two different chemistries at the same time. And so it's often very difficult, but you saw that with layer by layer, I can mix and match chemistries to do two things at the same time. Paula showed the same thing, of course, with her drug delivery. You can compartmentalize things, you can put multiple components in. It's very mix and match.
Got it, I understand. So Paula, kind of along those lines, I'm going to go back to you. And I think the answer is likely yes, but maybe you can expand a little bit on, the question from Delwyn is, can these LVL films that you described and Jamie describes really be deposited as conformal coatings on pattern surfaces? And I'm guessing kind of macroscopic pattern surfaces, yes, but when the dimensions get small, does that cause any particular challenges?
Yes, so actually there's been some really nice work done by several groups which show the kinds of features that you can coat with layer by layer films. And resolution can be as low as sub-micron quite easily. In some of the work that we've done, we've actually coated microneedle arrays very conformally. There have been very complex grid patterns. You saw in Jamie's images that the contours of fabrics are replicated. And layer by layer films have even coated things as intricate as virus bodies. Okay. However, when you get down to nanoscopic dimensions, as you begin to increase the number of layers, it's been shown that when you have short gaps that are sub-micron, you can begin to see some bridging effects depending on the molecular weight of the system that you're using and the number of layers that you're putting down.
Got it, understood. This is a little bit of, I don't think you talked about this, Paula, but it seemed to be something that maybe LBL could play a role, and this is from Daniel. It says, what specific LBL systems might be either applicable or been shown to be used for antibiotics, so to combat multi-drug resistance? Has there been work in that area that you know of?
Yes, so actually, layer by layer films can be designed to incorporate not just one, but multiple antibiotics. And this is one way of approaching multi-drug resistant species. But there's been more recent efforts to generate layer by layer film surfaces, which are microbicidal, and through their killing action of actually disrupting the cell membrane, are not susceptible to resistance mechanisms. And in our lab, we did some work on that with a colleague, Alex Klebanoff, at MIT, using inalkylated polyethyleneamines. More recently, though, I think the idea of using layer by layer films to incorporate or encapsulate either novel polymers or novel drugs which address some of the functions of bacteria that are less mutable can lead to ways to address multi-drug resistance too.
I see. Thank you. Jing Shen, Jamie, and there's a second question also along these lines, Jamie, for you, is UV grafting or cross-linking anytime necessary to get these coatings, or do you have anything more to say about how the action of UV light and some reactive species in LBLs could be applied?
Certainly, depending on what property you're talking about and what you're after, many of these you saw both with Paula's work and my work, you saw that the polyelectrolytes themselves have a lot of functional handles. You know, you have NH2 groups and other sorts of things hanging off of these polymers, and of course they would lend themselves to various types of cross-linking, including UV if you use the appropriate chemistry. And you could, for example, perhaps improve robustness. I know people are interested in wash durability of textile treatments, and certainly cross-linking with UV is one option to impart that kind of durability, if you will.
I see. This is from Alec Gapan. What about moisture barrier? Now, a lot of these things come from water in the deposition, but could you say a little bit more about any potential applications in moisture barriers?
Yeah, so moisture barriers is clearly a challenge because we are working with water-based ingredients, and you end up with a coating typically that is hydrophilic. Certainly a number of layer-by-layer groups have made super hydrophobic surfaces, but even a super hydrophobic surface isn't necessarily a barrier to molecular water. But I used vermiculite clay in one of my examples, and a vermiculite clay coating, because the vermiculite is not very hydrophilic, it's hydrophilic but not super hydrophilic, and it has a big aspect ratio, you can get some amount of moisture barrier, but I get 1000x improvement on PET with oxygen, and I might only get a 3x improvement with moisture. And so the moisture is absolutely a challenge. Certainly layer by layer could be combined with other technologies as well to get both the moisture and the oxygen, including lamination and other things. There's a lot of tricks you could play.
Jamie, I'm going to have the last question here for you before we wrap up. I got a question here about the flame retardant, or I guess other types of materials, but flame retardant material getting washed away. And is the LBL assembly pH reversible? So that kind of combines a couple things there. How robust are these things to what you might expect them to receive in maybe a typical cleaning cycle?
Okay, so for the wash durability more broadly, I think you would certainly cross-link the systems I showed. They're not going to be extremely wash resistant under the conditions because you have a very high pH in the washing machine with detergent. But we do know that cross-linking then can secure those coatings. And this would be true of almost any coating you would put on fibers. I know that I can bring Paula into this discussion because I know she's worked on drug delivery with pH response. And so, of course, some of that pH response is disassembling the film. In that case, for drug delivery, it's a good thing. You want it to disassemble. So it just depends what you're after. If you don't want something to disassemble, you would certainly cross-link it. If you use strong polyelectrolytes, it's also much more resistant to pH than a weak polyelectrolyte. But all of this depends on the exact chemistry you're using and what exact pH level you're talking about.
Paula, did you want to follow on that?
Yes, absolutely. Jamie's really said it spot on. I think if you're trying to create something extremely stable, you work with strong polyelectrolytes or with polyelectrolytes that can be cross-linked. If you want something that is going to be pH responsive intentionally, you will choose something like a polymer that will undergo some sort of transition over the pH range you're interested in. And that's why the acids become interesting for us for drug delivery and some of the polyamines.
Well, thank you, Paula. Thank you, Jamie. This was an excellent webinar series. I have a feeling this won't be your last poly webinar, but I'll contact you later about that. Both of you, thank you very much for doing this. Thank you to all the attendees for joining us for this poly webinar, and look forward to a future poly webinar sometime later this fall. But don't sign off yet. Let me turn it back over to Mike for some closing comments.
We wanted to let you know that if you enjoyed today's discussion, there are all the previous Division of Polymer Chemistry webinars, which Eric just pushed through on the chat window, so you can click on any of those links. Or you can go to YouTube and simply search for the Division of Polymer Chemistry to find their channel. There are many benefits to being a member of the Division of Polymer Chemistry, including access to technical programming, discounts on workshops, and much more. Remember that the first year is free, and you can reach out to Kathy Mitchum, whose email is provided here, to find out more information. And that wraps up our program for today. So thank you so much for joining us, and we will see you in November.